Food allergies can trigger swift and sometimes life-threatening reactions. And new research is shedding light on how exactly these dangerous responses are initiated. But here's where it gets controversial. What if we're just scratching the surface of what truly causes anaphylaxis? Recent studies involving mice have uncovered surprising clues, indicating a specific process in the gut that kicks off severe allergic reactions. And these insights could point us toward new ways to prevent them. Anaphylaxis is a rapid-onset allergic response that can turn deadly within minutes. It is commonly brought on by insect stings, certain medications, or foods like peanuts and eggs. When someone with a food allergy encounters their trigger, their immune system can overreact to the presence of the allergen. This overreaction causes symptoms like swelling, difficulty breathing, and dangerously low blood pressure, which, if not treated promptly, can be life-threatening. Currently, the main treatment for anaphylaxis is epinephrine, administered via injection or, starting in 2024, a nasal spray. This hormone helps by relaxing constricted airways and constricting blood vessels to stabilize the patient. Although it saves lives, epinephrine is only a remedy, not a preventative solution. As immunologist Tamara Hack from Indiana University highlights, epinephrine only works once the reaction has already started. So we desperately need methods to stop anaphylaxis from occurring in the first place. The recent mouse studies focus on understanding what happens at the earliest moments of allergen exposure. Researchers found a crucial molecular signal in the gut, Leukotrienes, that appear to play a central role in activating the cascade leading to anaphylaxis. In simplified terms, edible bits navigate through the intestinal lining into the bloodstream, a process that can trigger mast cells, the immune system's sentinels that recognize perceived threats and unleash allergic responses. The key discovery here is that leukotrienes regulate the transportation of food particles across the intestinal barrier. Stephanie Eisenbarth, an immunologist from Northwestern University who contributed to one of these studies, explains, once we grasped how this pathway functions, it became clear that blocking it could prevent the entire chain of events. If there's no ferryboat crossing the allergen into the bloodstream, then mast cells won't detect the threat, and the allergic reaction won't start. This insight led researchers to test a drug called Zileuton, already approved for asthma management, which successfully diminished allergic responses in mice exposed to peanuts and eggs. It's important to note that these findings are specific to food allergens that reach the gut. Different mechanisms seem to be at play with insect stings or other airborne allergens. For example, Nathaniel Bachtel from Yale, another co-author, points out that injected allergens didn't rely on leukotrienes in the same way, indicating distinct pathways for different types of allergic triggers. The studies also explored the behavior of specialized mast cells in the gut lining, providing a more detailed picture of the immune response. The process by which allergens trigger reactions is far from fully understood, and this complexity is why these discoveries are so exciting. They shed light on a part of the immune response that has been overlooked for too long. Both studies emphasize leukotrienes as promising targets for future research and potential preventative treatments. Bachtel remarks, "It's somewhat surprising that this pathway hadn't been examined in such detail before." Although these findings come from animal models, there's strong reason to believe humans share similar mechanisms. Hack notes, the data strongly suggest that testing these pathways in humans is justified, and ongoing clinical trials are already being planned. Co-author Adam Williams explains that the initial human studies will focus on whether Zileuton can influence how food particles pass through the human gut, specifically in people with various food allergies, excluding peanuts at first and more controversially, Do these findings challenge our current understanding of allergy triggers? Share your thoughts. Do you believe this new pathway could revolutionize allergy prevention? Or is there more to uncover?